Okay, so let's start by drawing a cell, and we're going to make this one nice and big so it's easy to see what's inside the cell. So what we're drawing here is called the cell membrane, and cell membranes surround every cell, whether it's eukaryotic or prokaryotic, whether it's an animal cell or whatnot. Next, we're going to draw nucleus, nice and big, and that's the control center of the cell. And inside a nucleus, there's a very dark region called the nucleolus. So the nucleus contains the instructions for how to make things. The nucleolus makes these little things called ribosomes, which are like little machines inside the cell, and they're in charge of putting things together, making stuff. So what we're drawing here is a series of tunnels and hallways, and that is going to be the rough endoplasmic reticulum, or the rough ER. And we know it's rough because we can see all those dots on it. Those dots are the ribosomes. If there are no dots on that series of hallways attached to the nucleus, then it's smooth ER. So here we're labeling this guy rough because it has ribosomes on it, and here we're labeling this one smooth because there are no ribosomes. Either way, the ER is attached to the nucleus. It's a hallway transportation center attached directly to the nucleus. And there we are labeling the ribosomes. Now you'll notice there are ribosomes also throughout the whole cell. They're not only attached to the rough ER, they can be located floating around in the cell in the cytosol, which is the jelly-like substance. So what happens is the instructions inside the nucleus come out, a copy of the instructions into the rough ER, go to the ribosomes. The ribosomes are in charge of putting things together. So when the ribosomes are done assembling their product, they put whatever they've made into these little containers called vesicles. And you'll see the arrows there show the direction of the vesicles. So the vesicles is literally like a cup or a bowl or a lunch tray delivering items throughout the cell. So here we see the vesicles delivering items to a series of stacked membranes. Those are called the Golgi bodies. We'll label those in a minute. Now the Golgi bodies, what they do is they do a couple things. They check to make sure that the contents of the vesicles is correct. And if it is correct, then they package them up in vesicles and send them off throughout the cell or out of the cell. If the product that the ribosomes, the little machines, made is not good, if they find an error, what the Golgi bodies do is they mark it and target those products for destruction. And it sends the vesicles to another large container called the lysosome. And what the lysosomes do is they contain digestive enzymes, and they break apart the products that were in the vesicles back to basics, and basically the lysosomes recycle the products that were made badly. But it all starts with the instructions that are located inside the nucleus, and we call those instructions DNA, deoxyribonucleic acids, and we'll learn about them shortly.